This video is about using sorghum or sorghum Sudan grass for managing soil health. Leguminous cover crops such as sun hemp are efficient in fixing nitrogen and making it available to cash crops. However, the residues of these cover crops generally have low carbon to nitrogen ratios and will break down quickly, thus contribute little to adding to soil organic matter, which is the backbone of building a healthy soil structure, i.e., improving water holding capacity, aggregate stability, and water infiltration. Grassy cover crops such as sorghum with a high carbon to nitrogen ratio would break down slowly, accumulate more dry biomass, transpire less water, decompose slower, and thus contribute more to soil organic matter. In addition, the robust root system of cover crops like sorghum can also scavenge nutrients leached into the soil, preventing soil nutrient losses. Sorghum has a high biomass and adds organic matter, which contributes to soil building. The crop also has a deep root system, which adds to its drought tolerance and ability to scavenge soil nutrients. It is also tolerant to a wide range of pHs and contains durin, which can be converted to biofumigants used against soil-borne pathogens, and sorgolone, which suppresses weeds. As if there weren't enough reasons to incorporate sorghum to your rotation, the crop can also be used for forage, grain, or energy. Here you can see the 5 varieties of sorghum used in the trial and their individual growth habits. Biomass production of different varieties varied significantly at 2.5 months after planting. NX2, which is an energy sorghum, produced the most biomass, whereas forage sorghum and cow vittle produced the least. This may affect the water conservation capabilities of these cover crops. This project will evaluate the water use efficiency, or WUE, of sorghum in a conservation tillage agro-ecosystem. This video shows the termination of the sorghum with the BCS-operated flail mower. The above-ground biomass drops behind the mower and is left as a surface organic mulch. Although water can be lost through evapotranspiration while the sorghum is growing, evapotranspiration was reduced immediately after mowing when compared to soil under bare fallow. This reduction in evapotranspiration likely will last even after mowing. Eggplants were planted into the surface organic mulch of different sorghum and grown for 5 months. Higher biomass resulted in higher soil moisture 4 months into eggplant planting. All field plots that had sorghum surface organic mulch had higher soil moisture content throughout the eggplant growing period. The organic mulch reduced water transpiration from the soil surface and led to higher moisture retention. Underneath the surface of the organic mulch, an abundance of white mold can be seen. The sorghum treatments had higher fungal biomass than the bare ground control. The robust root hairs of sorghum, sticky glomalin of mycorrhizal hyphae, actinomycete hyphae, along with polysaccharides of bacterial colonies clinging to the soil particles under the sorghum mulch, may have resulted in improved aggregate stability of the soil and led to better soil microporosity compared to bare ground treatment. Water infiltration rates from each plot were estimated using the double ring infiltration method. Sorghum did not improve water infiltration within one cropping cycle, which could be attributed to there not being enough time to change the macroporosity of the soil. However, since water holding capacity was increased by many of the sorghum treatments, the microporosity of the soil in the sorghum plots may have been increased and have led to better water holding capacity. Soil samples were collected from the sorghum mulch plots as well as from the bare ground control plot using the stainless steel soil cutter from the NRCS Expo rainfall simulation kit. A simulated rainfall experiment was done to compare water infiltration between the bare ground control and sorghum varieties. Immediately after the simulation, which mimicked torrential downfall, the bare ground treatment experienced the most water runoff and soil sediment, which would likely result in soil erosion under real-life conditions. Among the treatments, NX2 sorghum experienced minimal to no runoff and highest infiltration rates into the bottom of the soil profile. Under real-life conditions, this water that infiltrated could lead to the recharging of aquifers. Bare ground had the highest water runoff sediment, and percolated sediment, indicating higher soil erosion due to poor soil structure. On the other hand, water infiltration was highest in NX2 and lowest in the control group. All sorghum treatments performed better than the control. All the sorghum treatments had white mold colonizing the soil surface, but bare ground did not. Mycelium growth contributes to better soil aggregation, hence better water percolation. Discrepancy between the infiltration results demonstrated here with the field double ring infiltration could be because the surface organic mulch did not affect the lower soil profile. In this sorghum conservation tillage system, we can see clear effects of using sorghum as a cover crop. NX2 was especially effective in improving water conservation properties. However, without tilling sorghum into the soil, we did not observe suppression of plant parasitic nematodes by any of the sorghum varieties tested. Interestingly, a negative relationship between all the soil properties related to water conservation and root knot nematode abundance in the soil were observed. Further research will be conducted to examine the plant parasitic nematode suppressive as well as water conservation potential of sorghum in a strip till cover crop systems. This project is in part supported by Western SARE and NRCS.